I'm Petra Sakaris, and I'm taking environmental science with a concentration in biology at Carleton University. My thesis will be conducted in the Bahamas, and I'll be studying the sublethal effects of cold shock on bonefish under the supervision of Dr. Stephen Cook. This is my species of interest, the bonefish. Bonefish live in tidal flats and are called the ghosts of the flats because of their ability to blend into their surroundings. First, let me define cold shock for you. Cold shock is when a fish experiences temperatures that are below ambient for an extended period of time. Ambient just means normal conditions, so in my case, it means normal temperature, which is around 25 degrees Celsius in the Bahamas. Bonefish have a huge economic impact in the Bahamas. In 2010 alone, bone fishing brought in approximately $140 million. They are also ecologically important in nutrient dispersion and are an indicator of ecosystem health in tidal flats like the one you see behind me. You may be thinking that understanding the effects of cold shock isn't an obvious concern for a tropical fish species. However, events like the 2010 Florida cold snap have really got the scientific community thinking about the impacts these events have on surviving fish. The fish here in the middle is a young bonefish. To my knowledge, there hasn't been any previous research done on cold shock in bonefish. My research question is, does a cold shock event cause behavioral or physiological changes in bonefish? And if so, does a larger decrease in temperature cause more of an impairment to the fish? So, I'm hoping that my research will contribute to the field of fish conservation by providing baseline information on the physiological and behavioral impacts of cold shock on bonefish. Now for a short outline of what my methods will be. My data collection will be happening at the Cape Eleuthera Institute in the Bahamas between April and June. I will have 4 treatment groups: a control group, a handled control group, a 7-degree drop from ambient group, and lastly a 14-degree drop from ambient group. In total, I will be collecting data from 32 bonefish. Throughout my experiment, I will be checking ventilation rates and the fish's reflex response. In short, the experiment will consist of a 2-hour cold shock exposure time for the 7-degree drop and the 14-degree drop, with blood sampling directly after. The fish will then be allowed to rest for 2 hours in ambient water. After the rest, I'm going to conduct a chase to exhaustion, which examines the bonefish's response to an attacking predator. The ecological importance of this is determining how a fish responds to an attack after a short cold shock event. So for instance, are fish that experience colder temperatures more likely to be predated on because of swimming slowly or because of being easily exhausted? These are the types of questions I'm interested in. The bonefish are then going to rest in ambient water for another 2 hours. There will be another blood sample taken and the fish will be measured and tagged before being released. It's very exciting to be part of research like this through Carleton University, and I can't wait to see what the results of my data collection will be. Please contact me at this email address if you have any questions. Thanks for watching.